I cannot stress enough how helpful it is to know prefixes, word roots, and suffixes that you're commonly going to see put together in different ways to form a bunch of jargon that'll seem really scary, but you can really figure out what it means. And so these are some of the common word parts that I recommend that you learn and remember and use to your advantage to figure out what things mean when they might seem like big scary words. If you know the word roots and you know prefixes and suffixes, you can actually figure out a lot of what things probably mean, and a lot of times you're right. So you'll definitely want to know your metric prefixes, but then you're also going to want to know mono. So that's a single or one. The monomer is a single unit, so it's a molecule acting by itself. We often use this term when we're talking about proteins that maybe are multimers, so there's lots of copies. If you have a single subunit or a single molecule acting by itself, we see this term monomer. You'll then see bi and di, which both mean two, tri, which is three, tetr, quartr, four, pent, hex, sept, oct, non, deci. So you'll definitely want to know those. And then we have those words that, rather than a specific number, it refers to a sort of little of something, some of something, a lot of something. And so oligo means few or little. So if you talk about an oligonucleotide, that's the short nucleic acid chain, such as a PCR primer. If you talk about an oligopeptide, that's a short chain of amino acids. And then once those chains get longer, we refer to them as poly. And so poly means many. So a polymer is a long chain of linked together monomers. And some of the common polymers that we see in biochemistry include polypeptides, which are long chains of amino acids, which then fold up to give you a protein. You also see the prefix multi or multi, so multiple. And so a multimer is a word that's typically used to refer to a protein that has multiple subunits or chains. And we'll see how we can further classify these as heteromeric and homomeric, and we can have dimers and trimers. Those would all be examples of multimers. Pleio, these mean more. So if you hear about something being pleiotropic, that means it's doing or affecting multiple things. And so sometimes this is a drug doing more than you want it to do. So if it has like off-target effects, if it has lots of effects, and maybe you're trying to tease apart those effects, you could refer to all of those as being pleiotropic effects. Hypo, think hypo below. And so hypo means under or below. So if something is hypoactive, it's less active than normal. And if something's hypotonic, it has a lower tonicity. Hyper, over or above. So remember hyper, over. Think about being hyperactive. People are really, really active, really excited. They're more active than normal. And so we can talk about hyperactive protein. So a mutation causes a change in the protein that makes it so it is, say, constitutively active, or so it's acting all the time and it's not subject to its regulation as it normally would be. We can also talk about hypertonic, something having a higher tonicity, thinking osmosis and things like this. If something is hypertonic, then we're going to have water flow into it. And if something is hypotonic, you're going to have water flow out of it. So hypo, below, hyper, over. Epi, this is another one that means over or above. Here you'll see it in things like epigenetics, which is the field dealing with genetic information that's above the level of the DNA sequence. And so this includes things like DNA modifications, histomethylation, things when you're talking about chromatin modifications. All of those would be examples of epigenetics. And the opposite way, we've got infra. So infra means below. So if you're talking about infrared, that would be wavelengths of light with frequencies below those of red light. And if you're talking about ultra, that's above. So ultraviolet, so wavelengths of light with frequencies above those of violet light. You see ultracentrifuge, so that's a centrifuge that spins at speeds above those of a normal centrifuge. So we have these multiple terms that kind of mean the same thing. So if we're talking about below, we're going to see hypo or infra. If we are talking about above, we'll see hyper, epi, or ultra, or supra. So supra also means beyond, over, or above. So if we're thinking about supramolecular, that means that it's made up of multiple molecules, not just one, but beyond one. Another term you'll see is meso, so middle or moderate. So something that's mesothermic means it's living at a moderate temperature. And so some organisms, say, are mesothermic, where they are adapted to live in a moderate temperature. Iso, equal or similar. So going back to tonicity, we will see the word isotonic. And so if something is isotonic, water is not going to flow in or out of it because it's equal tonicity to its surroundings. Isomers, these are molecules that are similar, but they differ in some way, so they maybe have different shapes. Those would be isomers. Ortho, this means normal, straight, or correct. So you'll see the term orthosteric. This means that something binds in the same place as the normal binding partner. And this is as opposed to allo, which means other. So if we're thinking about an enzyme, say, and it has a site where it's substrate, so where the thing that it normally binds and modifies, that it has that active site where that binds. And if something else binds there, that would be orthosteric binding. It would be binding to the same place as kind of that normal binding partner. But if something bound somewhere else on the enzyme, we would call that allo. And so an allosteric modulator or an allosteric binding partner would be binding at somewhere other than that normal active site. We also see this word allosteric effects, where basically something that's happening at one site has effects at another site. So allo, other. Ortho is kind of the normal, the straight, the correct. Endo and ento both mean inside. Think endo, inside, ento, inside. So if something's endogenous, that'd be something that's made by an organism rather than taken in from the outside, such as hormones that the body produces rather than hormones that you get from outside or a drug that you get from outside, the pharmaceutical drug. Those would be examples of exo or exogenous things. If you were giving them from the outside, that would be exogenous. So even if it was something that the body normally makes itself, if you give it from an external source, that would be an exogenous compound. So it's something that you kind of add artificially or from the outside. And you'll see exo or ecto. So think exo, external. And then you have your endo inside. So if you think about like an endoscopy, that's looking inside of you. Whereas if you think about like exoplanets or exoskeleton, those are things that are on the outside. And so you'll see them in terms like endogenous and exogenous. Auto, self. So an autotroph is an organism that's able to make its own food. Don't confuse inter and intra. So inter means between, whereas intra means within. So intermolecular interactions are interactions between different molecules, whereas intramolecular interactions are interactions between different parts of the same molecule. Although we often use the term like intermolecular forces, even when we're talking about intramolecular interactions, if it's a really big molecule. But intramolecular is something that's happening within the molecule itself, whereas intermolecular would be between different molecules. Trans and cis. So these are another pair of kind of opposites. Trans means across, whereas cis means same. So you'll see the word trans in things like transmembrane proteins. Those are proteins that go across or span membranes. And cis, this means same. So sometimes you might see like cis regulatory elements. These are regulatory non-coding regions of DNA, such as promoters that are on the same piece of DNA of a gene that regulate expression of that gene. So it's on the same piece of DNA as that gene, so we call it cis-regulatory. Whereas other times you'll have trans-regulatory elements that might be, say, proteins that are coming from a different place in the cell or something like that. Those would be trans-regulatory, whereas cis-regulatory would be things that were kind of on that same strand as the DNA and helping regulate it. Another pair of opposites, hetero and homo. So hetero means different or other. If something is heterogeneous, it's made up of multiple things or it's not consistently distributed. If something is a heteromer, it's a complex with different components. So we talked about how we could have like many. We had a multimer that had many subunits of like a protein with multiple chains, say. A heteromer would be that the different chains were not just copies of a single chain, but they were different actual chains. So maybe you have an alpha chain and a beta chain, and those come together as opposed to having two alpha chains. And the two alpha chains would be an example of a homomer, so a complex made with multiple copies of a single component. And similarly to how we had heterogeneous, which was made up of multiple things or not consistently distributed. If you think about like Rocky Road ice cream or something, as opposed to homogeneous, which is made up of a single thing and are consistently distributed. And so maybe you've got your vanilla ice cream where that vanilla is really nicely distributed throughout, or you only have a single component. Amphi, ampho, ambi. These all mean both. So amphoteric means something is able to act as both an acid and a base. Amphiphilic means having both hydrophilic and hydrophobic parts. So those prefixes all mean both. Per, this means through. So if something is permeable, it allows things through. Sometimes things will be semi-permeable, so they'll allow some things through, but not others. Thankfully, we've got that word semi, which means partly. Pan, this means all. So a pan-genome analysis is an experiment that looks across the entire genome, not just select parts. And if you think about like a panorama view, you're looking at all. So you're seeing all. So the pangenome analysis is looking across that entire genome, not just select parts. And if you see that prefix pan, think all. Holo, whole. So it sounds like whole, and it's holo. And so a holoenzyme would be an enzyme with any necessary cofactors bound. And so maybe that enzyme needs to bind to a metal or it needs to bind to a vitamin, something like that, in order to be able to function. If it's bound to that thing it needs, we call it the holoenzyme. If it doesn't have that thing it needs, or if it's without its binding partner, we call it an apoenzyme. And so we sometimes might talk about it doesn't have to be an enzyme. Sometimes it might just be a DNA binding protein or a protein that binds some sort of small molecule. If it doesn't have that small molecule, we call it apo. And if it does, we call it holo. And so if you're thinking about maybe you're doing something, looking at some structures of a protein with and without its cofactor, we would say that we have like the apo structure, where the protein's by itself, and then we have the holo structure where the protein's got its friends hanging with it. Neo, this means new. So gluconeogenesis is the making of new glucose from non-sugar parts. So basically making it new, you've got neo. Paleo, this is old. So now we've got kind of the opposite. We've got paleogenomics, which is the study of ancient DNA. So if you think about like a paleontologist who's studying fossils, that would be an example where you're seeing paleo. And you might see it with things like paleogenomics, which is the study of ancient DNA. Pro, this means before or in front of. So a prodrug would be an inactive version of a pharmaceutical compound that's then metabolized in the body to form an active compound. So maybe it has a chemical group removed or it's got something added, it's oxidized, it's reduced. Those things would be examples of you have the prodrug, which is then modified to form the active drug. And so you give it in the form of the prodrug, and then the body converts it into the active form. You also might see this in the case of a molecule that, say, isn't very soluble, it's not very stable, something like this. You'll add a group onto the drug before you give it, such as a phosphate group, and then that group that you added will allow the molecule to get where it needs to go, and then the body will convert it into the form that is active. And so you'd be giving a prodrug and giving an active drug. U means true, normal, good. So eukaryotes are organisms whose cells have a true nucleus, whereas pseudo means fake or false. So a pseudogene would be remnants of a gene that remain in the genome but aren't functional. Another pair of opposites, macro and micro. So macro is big. If you think about a Big Mac, big cheeseburger, macromolecules are big molecules. So they're large molecules, including nucleic acids and proteins and lipids. Things that are macroscopic, you can see with your naked eye, as opposed to things that are micro. So if you've got like a microscopic, it's something you need a microscope to see. So micro can mean small, or it can also mean more specifically 1 millionth. So a microliter is a millionth of a liter. In that case, you might also see it written with a mu symbol. Nano, so this means 1 billionth if we're thinking in numbers terms, but if we're thinking about how it's often used outside of a specific scientific context, you might see it just be used for super duper small, or people put it in the name to sound cool. Technically, a nano means a billionth, and so a nanometer would be a billionth of a meter. Nanodiscs are these tiny little lipid discs that you can use to surround membrane proteins to try to visualize the protein with things like cryo-EM. So that's the word nano. Nano is smaller than micro. It's 1,000 times smaller in the metric sense. And it also is micro and nano are smaller than macro, and macro means big and like kind of seeable. And if we're thinking about terms of microscopic versus microscopic. Co means with or together. So a co-precipitant is something like glycerol or glycoblue you add to help nucleic acids precipitate them and help draw them out of solution. Kin means move. So if you think about kinetics, that's the study of the rate of things, such as enzymatic reactions. A kinase is an enzyme that moves phosphate groups from ATP onto other molecules, well, typically ATP. Peri, this means around. So the periplasm is the space around the bacterial cytoplasm between the inner and outer membranes. Meta, this is beyond. So a metastasis is the spread of cancer beyond its original location. And telo is end. So telomeres are the end parts of chromosomes. Getting to some roots, gen or genesis, birth, creation, making. So glycogenesis is the making of glycogen, which is a storage form of sugar, which involves kind of linking those sugar molecules up together. Hydro means involving water or hydrogen. So hydrolysis is the breakdown of water or the breakdown of something else by water. Lipo or lipid, this involves lipids, so your fats, oils, waxes, those sorts of things. If you see the term lipophilic, that means lipid loving. And so a molecule that associates with lipids, such as membrane-bound proteins, we would describe that as lipophilic. Glycol or glyc, that involves sugars or sweet. So glycolysis is the breakdown of glucose or blood sugar. Glycoproteins are proteins that have sugar chains attached. In those cases, you're seeing this word glycol or glyc. Another word for involving sugars is saccharide. So a monosaccharide would be a sugar monomer. So remember that monomer would be a single unit, and a monosaccharide would be a single unit of sugar. So connecting our units, we've got mono and we've got saccharide. Halo. So this means involving salt. So a halophile would be an organism that lives in high salt conditions. Oh, as we'll see later when we get to the suffixes, if we think about philic, that means something that's kind of loves something. And so when we had a lipophilic, that would be lipid loving. And if we have halophiles, those would be, or halophilic, those would be liking salt. Oxy, those involve oxygen. So oxidative stress would be a type of cell stress that occurs due to the presence of high levels of reactive oxygen species. Basically, really energetic oxygen molecules that like to attack things. Sulfa, thio, mercapto, those are all terms that mean that something has sulfur involved. So dithiothreatol, or TTT, and beta-mercaptoethanol, BME, are a couple of commonly used reducing agents. So sulfur is really good at reducing things. And these terms both have different sulfur roots, but they both have roots that tell you that it has sulfur. So the thio or the mercapto. So we had oxygen, we had sulfur. If we see ferro, that means it's involving iron. So ferritin is a protein that binds and stores and transports iron. Ferroptosis is a form of iron-mediated cell death. And if we see the CuPr, it means it's involving copper, which you'll see in various chemical names. Cuprate, things like that. Heme is relating to blood. So you also might see like HAEM depending on what country you're in. Hematocytes, those would be blood cells. Therm, now we're involving heat. And so something that is mesothermic, so remember we saw mesothermic, where meso means kind of middle. And so a mesothermic thing would mean it would be living at moderate temperatures. The meso tells us that it's the middle, and then the therm tells us it involves heat. Another word that involves heat is calor. So calorimetry is where you measure changes in heat to study the energetics of molecular interactions and reactions. So therm and calor both tell you that something involves heat. Sometimes with heat, you also see light, and the prefix we use for light is color or chrome. So something that is colorimetric. So if you think about metric, measuring, color. So an experiment that has a colorimetric readout means that it's giving us a color-based readout, such as binding to a visible dye, or something changes the color, that would be colorimetric readout. Photo involving light. So photosynthesis is the making of food using energy from light. So photo light, color or chrome color, and therm or color heat. Now let's look into shapes and things. So stare or stereo, this involves your spatial orientation. So stereoisomers, so remember iso is different and stereo, well now we're involving spatial orientation. And so stereoisomers are chemicals that have the same atomic makeup in terms of what's linked to what, but they differ in the way that the groups stick out in space. Morph, this is shape, so morphology is the shape or study of shape. Hist or histo, this involves tissues, so histology is the study of tissues. So ology is study of, so histology is the study of tissues. Morph means shape, so morphology, so ology is study, so morphology is the shape or the study of shape. And histology would be with hist or histo, now we're involving tissues, same ology ending, and so now we're talking about with histology, we have the study of tissues. Site or site, that means cell, and so a cytoskeleton is a network of tubules, etc., that give cells their structure and help move things around. Erythrocyte, so erythra, that's red, and so we have erythrocyte is a red blood cell. You can also think about cytology, which would be the study of cells. Sometimes cells eat and drink, and so if you think about eat, that'd be phage or phago. And so phagocytosis, so phago, eat, cyte, cell, osis. Basically, we have the phagocytosis is a way in which cells can pinch their membrane in to take in things from the outside. So they're taking kind of a big bite, as opposed to penocytosis. Peno means drink, and so penocytosis is they're kind of just taking in a smaller portion of membranes. They're kind of like sipping the outside. You also see phage, so macro, remember that's big, and big eat is so a macrophage is an immune cell that eats foreign stuff. Four, this is barrier carry. So an ionophore is a molecule that helps transport ions across membranes. Dent, think about going to the dentist, you're thinking about biting or teeth. And so something that is tridentate, so tri, three, dent, bite or teeth. Something that is tridentate would be a chelating agent. So chel is going to be like claw, and so you've got a molecule, a chelating agent that bites down 3 times on metal. So it's got basically 3 lone pairs of electrons that can form these coordinate bonds to hold a metal in place. We call that tridentate. And now we're finally getting to the suffixes. And so I know we've introduced a couple of these before because we're using them, but you've got to go in some sort of order, and it seems logical to have suffixes last. And so let's talk about the suffixes now. So we talked about philic or phile, so that's loving or lover. So something that's hydrophilic is water loving or water loved. Lipophilic would be lipid loving or lipid loved. Halophile would be an organism that likes to grow in high salt conditions. And it's kind of opposite, it'd be phobic or phobe. So that means fearing or fearer. So something that's hydrophobic, the word sounds like it should mean water fearing or water feared, but more it's that the water avoids it because of the hydrophobic effect, and water wants to hang out with itself more than those other things. But we're not getting to that here. Phobic, phobe, you think fearing, fearer. Pathy means disease, so a tauopathy is a disease that's characterized by misfolded and aggregated tau protein. Cardiomyopathy, when you've got like heart problems and things, so pathy, think disease. Pathology, all that good stuff. Lysis means breaking or breakdown. So catalysis is the breaking down of chemicals for energy and or the building blocks. Hemolysis is the breakdown of blood cells. So heme, remember, was blood. Lysis breaking down. So we've got hemolysis or hemolysis, which is the breakdown of blood cells. If you see something that ends in ase, that's typically denoting an enzyme. So a nuclease, say, would be an enzyme that cuts nucleic acids. So sometimes a lot of times the kind of root will be the thing that the enzyme is acting on, but not always. And sometimes it'll be what the enzyme is breaking down. So like a nuclease would be cutting down and breaking down of nucleic acids, so DNA or RNA, whereas a protease would be breaking down protein. Omics, this is an experimental method or field that deals with gathering and analyzing huge amounts of data. So this has kind of been more of like a buzz thing in recent years, but omics, we have all this capacity to get a lot of data and analyze that data. And if we want to think about those kind of fields that do that, we would put the term omics. So proteomics is the study of all proteins. So remember, proteo is proteins. And so the study of like all the proteins that are made and how this changes under different circumstances or in different tissues. You'll see techniques like mass spectrometry used for proteomics a lot. Genomics, this would be the study and comparison of genomes. Maybe you're even looking at some of those epigenetic stuff. And so remember, epi was like above, and so it was above the genome. So you might even see the term epigenomics. See the term transcriptomics if you're studying RNA transcripts, that sort of thing. Okay, so we talked about a couple of eating terms. And so we had phage or phago, which was eat, and pino, which was drink. We also have this suffix troph or trophy or trophism, which tells you that something has to do with eating, nutrition, or growth. So an autotroph, remember auto is self, troph is eating, and so an autotroph is an organism that makes its own food. An organism that eats itself, but yes. Okay, graph. This is a device for measuring. Graph, that'd be a device for recording. So, like a chromatograph is a device that separates components based on their properties for detection and or characterization. You'll see often chromatographs will give you chromatograms. And so when you see the term gram, that's a chart or a graph. And so a chromatogram, remember chrome means color. So a chromatogram would be like a graph showing light absorbance corresponding to sample coming off of a chromatography column. And so that is that. Metry or meter, measurements. So mass spectrometry would be a technique that measures the mass to charge ratio of molecules. And spect, this is often I'll be telling you about like light, but spectra means like a spectrum. And so you've got a spectrum of sizes of the M to Z ratios and stuff with mass spectrometry. UGI, we talked about this like LUGI or UGI, those would be the study of. So biology would be the study of light. Stasis, this means you're standing or placed or staying. So homeostasis, so home, remember that's kind of like same, and homeostasis would be a state of equilibrium or balance where things stay the same. And we saw this one too, OS. So OSE, like with glucose, OSE, this OS ending would be like a sugar. So glucose, fructose. Don't confuse that with ACE. So remember, ASC was going to tell you something was an enzyme. OSE is going to tell you it's a sugar. And 8, this would be a suffix that you'll see for an anionic, or that is negatively charged molecule. So like a carboxylate would be the conjugate base of carboxylic acid. So that carboxylic acid gives up its proton and becomes its conjugate base. And we refer to that conjugate base then as a carboxylate. You also see like glutamic acid, glutamate, those sorts of things. That 8 is tipping you off that you have some sort of anion. So, if you know the word roots and you know prefixes and suffixes, you can actually figure out a lot of what things probably mean, and a lot of times you're right. And so these are really, really helpful to get to know, and I cannot stress enough how helpful it is to know these words, or at least most of them. Some of them you don't see as much, but some of them you'll see a lot and are going to be really, really helpful. So, hope they are.